Hello, dear students. Am I audible? Please let me know if I'm audible. So, on behalf of Intelletics, I welcome you all for the understanding, for the explanation of 10th standard ICSE biology as per the need of the hour, I can say, because you guys of 10th standard are going to appear for the board exams of bio on 15th. So here we start with the chapter that is structure of chromosomes, cell cycle and cell division. So let us understand about the structure of chromosomes. Now what do we mean by the cell cycle? So please understand students what exactly is meant by a cell cycle. See, whenever you say in science, whenever you say it's a cyclic change, it means it's a change which is repeated again and again after a definite time interval. So these are the changes that do take place again and again after a definite time interval. And this is in the life cycle of a cell. And this is the reason why you would call it as the cell cycle. So all the changes that do happen in a cell and which changes which repeat themselves after a definite time interval in a living cell, I shall say. So that entire thing is called as the cell cycle. So cell cycle includes all the changes occurring in the life cycle of a dividing cell. So these things have to be kept in mind. So please remember what exactly do we mean by the cell cycle. Now, going ahead, so cell division is the cell division is one of the most fundamental characteristics of life. This is a method which enables the life to perpetuate generation after generation. Here, perpetuation is nothing but continuation. Cell division helps in continuity of life. Okay, this is equally true in the case of the simplest organisms like amoeba, as is in the highly complex ones, such as ourselves, the giant sized elephants, or the tall coconut tree. Here, what it means is that cell division is not a property limited only to the simplest organisms, yeah, unicellular organisms, but this property is seen in all the living organisms. Please remember when the cell stops dividing, okay, when the cell does not divide, it simply means that it is no more existing. Okay, in the lower organisms, cell division can be a means of reproduction. I mean, in case of unicellular organisms. Students, it's a request to you if you have any doubts anywhere in any of the terms or terminologies, you can put it up in the chat box. Even though there is a lag, I shall still get back to you and clarify your doubts. So some of the details of cell division are slightly difficult to understand, but we have tried to make it as simple as possible. So, structure of chromosomes. Now, before we go to structure of chromosomes, it is important for anybody to know what exactly do you mean by chromosomes. Okay, so chromosomes, you have to remember, are thread-like structures present in the nucleus and which are the carriers of genes. So genes are located on the chromosomes. Okay, and genes are important because genes carry the genetic information in the form of DNA molecules. So you have learned earlier that the most conspicuous events occurring during cell divisions are all related to chromosomes. The duplicated chromosomes. Now, what is meant by duplicated chromosomes? That is, they form their own copy. Try to understand when one cell produces two or more daughter cells. Now, why did I say two or more daughter cells? Because in mitosis, there are only two daughter cells that are formed, whereas in case of meiosis, four daughter cells are formed. So, whenever one cell gives rise to another cell, okay, at that time, all the cell organelles and the entire matter present inside the cell has to be equally distributed among the daughter cells. This also includes the genetic matter, and this is the real reason why the chromosomes have to duplicate much before the cell division starts. So the duplicated chromosomes get evenly distributed into the daughter cells during mitosis. Friends, please remember mitosis and meiosis are the most important kinds of cell division that are seen in all the organisms. Okay, meiosis won't be seen in the lower organisms, of course, but mitosis and meiosis are the most common types of cell division you can remember. Thus, all body cells that are the result of a long chain of repeated mitotic divisions have the same type of chromosomes in the same number. This ensures the normal functioning of the cells and through that the life of the organism. Please remember whatever we have in our life, or rather whichever characters that we show, all each and every character is controlled by the genes that we possess. और ये genes कहाँ पर है? तो these genes are located on the chromosomes. So chromosomes are obviously important. Now remember that the number of chromosomes may differ from one organism to another, and the number of chromosomes always remains fixed for a particular organism. So what are chromosomes? When a normal unstained living cell is observed under a light microscope, its nucleus may not appear to contain any particular thing inside. Okay, why? Just because they are very, very minute, so they won't be seen. So nobody would understand what exactly the nucleus holds inside it. But when the same cell is stained with suitable dyes, dyes are nothing but the colored pigments, several structures become noticeable in the nucleus. In all probability, you may be looking at a non-dividing stage, that is interphase of the cell. Although depending upon the part of the body from where the cell has been taken, it may as well show some division stages too. Now please remember, when the cell is not in its dividing stage, we say it is in the interphase. So interphase is the phase in which the cell doesn't appear to be in its dividing stage. So this can be asked to you. The cell in the non-dividing stage is said to be in dash. Please remember, the non-dividing stage of the cell is nothing but the interphase. In the interphase, the nucleus shows a network of very long, extremely thin, dark staining fibers called as chromatin fibers. Friends, when the scientists did try to search inside the cell, did try to observe the cell under the microscope, very first they couldn't really observe anything. They couldn't really observe anything. The real reason behind it is because the cells are colorless. So they thought of using a stain. Now, stain is nothing but what? A dye or a colored pigment. And on using it, they found that the very first cell organelle that became very prominently visible were these chromosomes. And this is the reason why they call it as chromosomes. That is, chroma standing for colored and soma means body. That is, they called it as colored bodies. And the same we now call them as chromosomes. As the cell begins to enter the first stage, that is prophase of cell division, the chromatin fibers condense to form chromosomes. Very, very important point. Please keep this in mind. That is, आप जब cells को देखेंगे, तो cell कौन से stage में है, depending on that, you will find the various phases of the chromatin network. अगर आपको एक नेटवर्क दिखाई दे जहां पर आप कितने धागे जैसे स्ट्रक्चर्स है ये गिन नहीं पाते, you find that it's all in a complex network when you can't really count the number of fibers present in it. This means it is in the interphase. And as soon as the cell would enter the prophase, what happens is this chromatin network, it starts undergoing condensation. So as a result of condensation, the chromatin network then resolves, it becomes distinct, it starts separating from each other, and this results in the formation of definite fine thread-like structures which are called as chromosomes. So friends, please remember, अगर आपको कोई डायग्राम दिखाएंगे अगर एग्जाम्स में और कहा जाएगा find out which stage of the cell division it is. So if you can't really count the number of thread-like things, so it clearly indicates it's a chromatin network. So it is the interphase. Please, please, please keep this in mind. The chromosomes readily pick up certain dyes and get colored, and hence the name chromosomes. The word chroma means colored and soma means body. So how would you define chromosomes? So the chromosomes are the highly condensed coiled chromatin fibers. Any doubt with this particular paragraph? Sure. I know there is a lag. That's the reason I'm giving you a moment so that if you have any doubt, you can certainly type out your questions and I shall answer you. Coming to the next, that is 2.2, that is discovery of chromosomes. Chromosomes were first discovered in 1882 by a German scientist, Walter Fleming. Very, very important point. I shall highlight it. I hope you can see the highlighted area so that you remember this. Very, very important. So chromosomes were first discovered by German scientist Walter Fleming. This can be asked to you in an MCQ. He noticed these in the rapidly dividing cells of the larvae of salamander. Again, an important point I can ask you. In what did he first observe the chromosomes? So he had seen the chromosomes for the first time in the larvae of salamander, that is an amphibian for your information. The microscope he used was of the old type. Okay, so the microscope that he used was of the old type. Now, old type of microscope का मतलब क्या है यहाँ पर कि that was not that advanced. So it was a simple lens sort of a thing, simple magnifying lens sort of a thing. The microscope he used was of the old type, and through it he saw minute threads that appeared to be dividing lengthwise. Fleming called their division as mitosis because mitosis literally means this. Basically, in the word osis means condition and mito means thread. So please remember mitosis is nothing but a thread-like structure that is observed. Now, subsequently, with the help of more powerful microscopes and by using special techniques, the chromosomes and their constituents have been studied in great detail. So now let's understand the structure of chromosomes. Each chromosome in its condensed form, as visible during the start of cell division, consists of two chromatids joined at some point along the length. This point of attachment is called as a centromere and appears as a small constricted region. Please remember, chromatids are formed only and only after the interphase. This is because in the interphase, in the S phase, the DNA replicates. Now, when I say DNA replicates, it means it forms its own copy. And this is the reason why the chromosome, which normally is a single thread-like structure, now instead of a single thread-like thing, it consists of two thread-like structures. The reason is because on the other end, it has its duplicate of DNA. And these two thread-like structures, which carry the same genetic information, are called as the sister chromatids. And these remain connected with each other at the point that is centromere. And this centromere, when you observe the chromosome, it appears as a small constricted area, that is a furrow-like thing, a pinch thing, something which goes a bit narrow, deeper inside. The centromere also serves to attach to the spindle fiber during cell division. So please remember this as well. I can ask you, what is the function done by the centromere? So the centromere helps in attachment to the spindle fiber during cell division. Each chromosome centromere is located at a particular site. This means that every chromosome does not have its centromere at the same position. As the spindle fibers contract, the sister chromatids are separated at the centromere and each is pulled away from the other towards the two poles of the dividing cell. That is stages D and E that you will study later. I shall tell you this happens during anaphase and telophase of cell division. After the completion of cell division, the chromatids, now called as chromosomes, decondense, that is the exact opposite to what has happened in the prophase, and revert to their very long and fine thread-like chromatin fibers. So there would be as many chromatin fibers inside the nucleus as the number of chromosomes that appear during cell division. So this point also is important. There will be many chromatin fibers inside the nucleus that are equal to the number of chromosomes that appear during cell division. Any doubt with this particular part? Please let me know. Now coming to the next, that is chromatin. The chromatin material that constitutes the fiber is formed of two substances, very, very important. DNA up to about 4% and histones, proteins about 60%. Let me tell you one additional point here, that the histones are basic proteins, and that's the real reason why they get attached to the DNA, that is deoxyribonucleic acid. DNA is acidic, so the basic proteins cling to it and they help in stabilizing the structure. So each chromatin consists of two parts, that is DNA almost 40% and histones almost 60%. Histones are a type of proteins which are basic in nature. Now coming to figure 2.3 given below is a highly diagrammatic representation of the structure of the chromatin fibers, showing the sequentially enlarged view of the DNA strand. So you can see cell. Inside the nucleus, there are chromosomes present, right? Chromosomes contain chromatin fibers, which is supercoiled. Okay, and this supercoiling is because of further, first is the coiling, and what coils is the nucleosomes. And what is a nucleosome? It is a structure that is formed by 8 type of histone proteins surrounding, this is surrounded by the DNA fiber. So this is the nucleosome, the rounded structures which were earlier called as the bead and the string model. Now, if you have any doubt, you can ask me any question that you have on this. I shall give you a moment to go through this. I know there is a big lag here, and this is the real reason why I'm giving you a minute. Any doubt? Any doubt? Okay, then we go ahead. Friends, if you have any doubt in any word, please feel free to ask me. And if because of the time lag here, if you can't really connect at the same point and your question really is not answered, you still can get in touch with us in the next session. The DNA strand winds around a core of 8 histone molecules. So this is the reason why it is called as histone octamer. The word octamer itself tells you it's octamer. So it's 8 histone molecules. This core can be imagined like a football, around which a long rope is wound with 1 or 2 loops. Each such complex is called as a nucleosome. So I can ask you what exactly it is. The nucleosome is the bead-like structure. It's a rounded structure, the core of which, the inner part of which, core is nothing but the inside, I can say. The inner part of which is made up of 8 histone molecules, and around it, 1/3 of DNA loop is seen. Even though if your textbook gives it as 1 or 2 loops, precisely it's 1/3 loop of DNA that is found around each nucleosome. Now remember the most important point again, a single human chromosome may have about 1 million nucleosomes. The entire chromatin fiber is coiled and supercoiled, something like the coils and the supercoils we see in a typical telephone call. I'm talking about the landlines, which we used to have it in a house once upon a time with the long coiled cord. So this coiling and supercoiling can be compared to it. Coming to the next, that is structure of DNA. The shape of the DNA molecule was studied by Rosalind Franklin in 1953, and the structure was finally worked out by and Crick. This is one very important. The structure was studied by Rosalind Franklin, and it was finally worked out by Watson and Crick in the same year. Now, what is the difference? The structure was studied by Franklin, but the 3D model of this DNA was worked out and given by Watson and Crick. And hence, the double helical structure of DNA is mostly known as Watson and Crick model of DNA. The DNA is a very large single molecule, and hence it is described as a macromolecule. Please remember the word macro is large. Also remember that macromolecules are a group of large molecules. It's not just DNA. Okay, so if at all they ask you if DNA is the only macromolecule, true or false? So it is false. There are many more macromolecules. Most common and the most large, one of the large I can say, is DNA. It is composed of two complementary strands. Now, complementary strands are nothing but the matching strands wound around each other in a double helix. Very important. So, it is composed of, I'm underlining this, highlighting this, two complementary strands wound around each other in a double helix. So, this is what is the diagram of DNA, the Watson and Crick model of DNA. Each single DNA strand, that is one side of it, is composed of repeating units, which are called as the nucleotides, which are made up of three components: the phosphate sugar, that is a pentose sugar arranged lengthwise, and a nitrogenous base attached to the sugar inwards. Okay, to the sugar inwards, which extends to join by a hydrogen bond. Okay, so the complementary nitrogenous bases from the other strand are going to be connected with each other by this arrangement. Thus, the two strands together make a ladder-like arrangement with the nitrogenous bases forming the rungs of the ladder. So imagine a ladder. The steps are nothing but the rungs. Okay, so the nitrogenous bases they form the rungs of the ladder. And the nitrogenous bases, why are they called so simple? They're called as the nitrogenous bases because they contain nitrogen. And the bases that are present, the nitrogenous bases that are present, are adenine, guanine, cytosine, and thymine, of which adenine pairs with thymine with two hydrogen bonds, and guanine pairs with cytosine with three hydrogen bonds. Very important. Adenine pairs with thymine with two hydrogen bonds, and cytosine pairs with guanine with three hydrogen bonds. So this is what is the structure. So friends, please remember, I shall give you a simple example so that it becomes a bit easier for you to have a clear understanding about what exactly the DNA strand is. For example, the place where which we live, that is our house, consists of walls. Now, the wall, if you break it open, it consists of bricks. If I crush the brick, it is made up of soil, right? So, in the similar way, imagine the wall, the entire wall, to be the DNA. If I break it open, it consists of smaller parts in it, which are called as the nucleotides. And if I break these nucleotides, what I get is a nitrogenous base, a sugar, and a phosphate. Please remember, nitrogenous bases are of two types, purines and pyrimidines. In your textbook, they have directly given you as adenine, guanine, cytosine, thymine, but ideally, they are classified as purines and pyrimidines. Okay, so purines and pyrimidines, which includes adenine, thymine, cytosine, guanine. So keep this in mind. Formation of the new DNA. See, when a cell will form its own copy, so the nucleus also has to be sent into both the daughter cells, and it should contain the same material that was present in the earlier, that is the parent cell. And for this reason, the DNA replicates, and as I have mentioned earlier, the DNA replicates in the S phase of the interphase. So S phase is the synthetic phase of the cell cycle, which includes, or which is important because it includes the replication of DNA as well as formation of the histone molecules. Please remember, histones are the basic proteins that support the structure of the DNA and help in the formation of chromosomes. Now, coming to the next, that is formation of the new DNA. During the interphase of the cell cycle, each DNA molecule, that is the chromosome, duplicates in readiness for their equitable distribution. Equitable is nothing but equal distribution in the two daughter cells during mitosis. For replication, friends, either I call it as replication or I can call it as duplication. That means generating its own replica or duplicate, in simple words, generating its exact copy, identical copy. That's the reason why we use the word replication. So for replication, the DNA double helix opens at one end, making the two strands free, to which the new strands begin to form, and the process continues in a sequence for the whole length of the DNA. The entire replication is a very complicated process which you will study in details in the higher classes. So friends, this is something that you will study in your 11th and 12th standard. You just have to remember that it forms its own copy for which it opens up, because if at all the two strands are connected with each other, in that case the DNA synthesis cannot take place. It can't replicate, it can't form its own copy, it can't be copied. Simple. Now coming to the next point, what are genes? Genes are specific sequences of nucleotides on a chromosome that encode particular proteins which express in the form of some particular feature of the body. Now please remember, genes are specific sequences of nucleotides. Genes are segments of DNA. And remember that each and every character that we possess is controlled by the genes. Okay, so how do the genes control our characters? The very simple thing is the genes sorry, contain the information in the coded form, and every character that we possess is controlled by a protein. So the formation of this protein is under the control of genes. So there are n number of types of proteins existing in nature, innumerable proteins, even though we have a definite number of amino acids, that is 20 amino acids existing in nature, and two more have been discovered. But till now we consider it as 20 amino acids. But the number of proteins those are present are just innumerable. And every protein need not have all the amino acids. Keep this in mind. Now, the extra point here, which is not in the syllabus, but still we'll read it because I can ask you an MCQ in this. Lots of regions in between the genes are just non-functional. Such areas, about 99% of the total DNA, show tremendous variations from person to person and help in identification of the individual by what is popularly known as DNA fingerprinting, but better called as DNA profiling. So friends, remember, DNA fingerprinting, that is DNA profiling, is a technique which can help us to identify an individual. Okay, so our identity is because of the DNA that we possess, and to establish, to find out this identity, we can conduct a process that is called as DNA fingerprinting, also called as DNA profiling. DNA fingerprinting is useful in establishing paternity and maternity in criminal cases. That's one of the applications of DNA fingerprinting. I said one of the applications. What I mean to say is there are many, many more applications of this process, that is DNA fingerprinting. Blood or semen stains, hair, or items of clothing found at the site of crime provided biological evidence through DNA fingerprinting. Okay, so any doubt with this? Any doubt? Any doubt, please let me know. I know there is a big time lag, and this is the reason why I'm just giving you a moment. If you have any doubts, you're free to ask me any doubts so that I can help you out. Okay, so coming to the next point, please remember blood or semen stains, hair, or items of clothing found at the site of the crime provide biological evidence through DNA fingerprinting. I can ask you, are the samples that can be used as evidence for establishing the identity? So this is it. So friends, going to the next page now. I'm just waiting for it to get downloaded. Now the next is cell division. Okay. So now let's continue with this. 2B, that is cell division, yeah, new cells from the existing ones. New cells need to be produced. Now let us understand why new cells are essential. Please remember every cell that is produced has its own lifetime, after which the cell obviously dies. Now when the cell dies, its functions, the functions performed by that cell, are going to be hampered. So instead, what the body does is, when the cell is about to die, before its death, the body ensures that it produces its own copy so that the functions of the cells are not affected. However, the rate of cell division is different from each cell in the body. It's different for each cell in the body. And this rate of cell division slightly decreases as we start aging up. Chirag Mehta, this chapter is all about cell division and chromosomes, which will be very useful for you. Okay, so if you have doubts, you can definitely ask us. That's not a problem. Now, so try to understand every cell has its own lifetime. So after that particular lifetime, the cell would die. And if this cell dies, its function is affected. You take an instance that if by chance, because of some reason, the few cells of the body they die, even though they were not really meant to die, but they die maybe because of an injury, because of the wear and tear. So in that case, their functions should not be affected. And this is the real reason why we need more and more cells to perform their functions. And this is how cell division is essential. Now, remember, cell division is essential for growth, for replacement. That is if there is any defective cell, it has to be replaced by a correct one. So that stands for replacement. Repair, as I said, if there is any injured area, the cell needs to repair itself. Okay, and reproduction. There are few cells in our body which also help in the process of reproduction. So this is the importance of cell division. First is growth, second is replacement, third is repair, and fourth is reduction. Now let's understand something about the first function, that is for growth. We all know that our life has started with a single cell. But the human body is made up of millions and millions of cells. So this is very sure that even though our life started up with a single cell and now we have millions of cells, this means the cells have undergone repeated division and which has resulted in the growth of the organism. So every organism, be it a plant or an animal, begins its life as a single cell. The single cell from which our life starts is the fertilized egg, which is also called as zygote. Please remember, zygote is the first cell of a life, which is also called as the fertilized egg, or fertilized egg is called as zygote, whichever way you want to remember it. This cell divides repeatedly to form a cluster of cells. Cluster is nothing but group of cells which start shaping for different functions to form tissues and organs. So cells come together to form tissues, tissues come together to form organs, organs come together to form organ systems, and organ systems come together to form an organism. Thus, cell division is essential for growth. Now, you can see this particular image. This is a 4-day-old human embryo which consists of just 16 cells, resulting from 4 cell divisions. Because as I said, our life had started out with a single cell that's called as a zygote or the fertilized egg. So it undergoes first cell division to form 2 cells, second cell division resulting in the formation of 4 cells, third cell division to form 8 cells, and fourth cell division to form 16 cells. As they multiply, they specialize to form the tissues and organs of the body. Any doubt with this? Any doubt with this? Now, coming to the next, that is point number 2 for replacement. There is always a wear and tear of cells during the normal body functions. For example, 20 million red blood cells, that is RBCs, in our body are destroyed every minute. A very, very important point. 20 million red blood cells in our body are destroyed every minute, and these are replaced by the new cells formed through a division of their parent cells in the bone marrow. So remember, RBCs are generated in bone marrow. Bone marrow is nothing but the hollow space that is present inside the long bones of a body where blood cells are generated. Not just RBCs, all the blood cells are produced inside the bone marrow itself. Now, in plants, the old and the dried leaves fall off and the new ones grow out. So that means there is a replacement. Now, coming to the next point for repair. Apart from normal wear and tear of the tissues in the body, there may be accidental injuries, as I did mention. One may get cuts in the skin or fractures in the bone. This is nothing but an injury. And at that time, what is essential is the repair. So, repair of such injuries is again through the cells which divide, which covers up the gaps and joins the broken ends. Remember, in cell divisions for growth, replacement, and repair, the number of chromosomes remains the same at each division. Remember, chromosome number never changes. So remember this, in cell divisions for growth, replacement, and repair, the number of chromosomes remains the same at each cell division. The chromosomes duplicate and distribute equally in the daughter cells. This kind of division, which occurs in all the body cells except in the egg producing oocytes, that is in ovaries and sperm producing cells, that is spermatocytes in the testis, it is known as mitosis. So ovaries, that is the cells, that those are oocytes, and the spermatocytes in the testes undergo meiosis and not mitosis. So keep this in mind. This cell division, that is mitosis, is seen in all the body cells other than the germ cells, that is the cells in the ovaries, the cells in the testes. They divide by a process which is called as meiosis. Any doubt with this? I shall give you a moment. Because of the lag, I keep saying this. So remember friends, ovaries, that is the cells in the ovaries, I mean to say the oocytes, and the spermatocytes, that is the cells in the testes, undergo meiosis. For all those who are wondering about this, please remember the testes and the ovaries are the part of the reproductive system of an animal. Plants may, we do not use this term. Please keep this in mind. These terms are applicable only for an animal body. Coming to the next, that is for reproduction. Reproduction is also through the activity of the dividing cells. Amoeba or bacteria just divide to produce two similar independent cells by mitosis. Remember, mitosis in case of amoeba or bacteria helps in reproduction. It's a unicellular organism. In higher forms, as in humans or in the banyan tree, special cells in the reproductive organs undergo a special kind of cell division that is called as meiosis to produce sperms and eggs. I've already mentioned, so keep this point in mind. In higher forms, as in humans or in the banyan tree, special cells in the reproductive organs undergo a special kind of cell division, which is called as meiosis, to produce sperms and eggs. The sperms and eggs receive only half the number of chromosomes of their parent cell. That is, the number of chromosomes of the parent cell, one chromosome for each egg. This reduction in chromosome number is very significant. Now try to understand. Human beings have got 46 chromosomes. So if two cells of 46 chromosomes each fuse together, the new organism that is produced would have got 92 chromosomes. Then 92 plus 92 would have been something else. So by this, the number of chromosomes till now would have reached millions. But this has not happened. This means that during meiosis, already there is a reduction in the number of chromosomes. So you can see this particular tabular form giving you an idea of the same. Body cells in man as well as in women have 46 chromosomes each. The sex cells, that is the sperms and the eggs, have only 23 chromosomes in them and they are represented as N. N stands for the number of chromosomes. The fertilized egg, that is from the sperm. Okay, fertilized egg. How is the egg fertilized? By the fusion of the sperm and the egg. So sperm consists of 23 chromosomes and the egg also consists of 23 chromosomes. So 23 plus 23 forms the first cell of the baby, that is zygote, consisting of 46 chromosomes, and that is what we call it as 23 pairs. Any doubt with this? Any doubt with this? Please ask me if you have any doubt. So remember, the sperms and the eggs contain 23 chromosomes each. There is no exception to this. Now let us talk about the next 2.6, that is types of cell division. So what are the types of cell division? There are two main types of cell division: mitosis and meiosis. In mitosis, the cell division results in growth and development. You can be asked the other way around. The cell division helps is dash. It is mitosis. Whereas the cell division that helps in production of is meiosis. Whereas mitosis helps in growth and development, whereas meiosis helps in the production of gametes. Now let us start talking about mitosis. The word mitosis comes from two words. Mitos, which means thread, referring to chromatin thread. Keep this in mind as well. The mitosis is a cell division in which one parent cell divides into two identical daughter cells. Remember, in meiosis, one cell produces four daughter cells, whereas in mitosis, one cell produces just two daughter cells. So friends, we shall take up a small break here and we shall continue from mitosis. Okay, so we shall take up a small break. Stay connected with us. Keep studying. If you have any doubts, please write them down in a notebook so that as soon as we come live for the live streaming in the next session, in which we will start with mitosis, you can certainly put up your questions there and I can clarify all your questions there, all your doubts. So friends, please stay connected. Also, we have to solve many practice sessions. I mean, we are going to conduct many practice sessions for doing the same, for solving up the MCQs and making you ready for the board. Till then, please ask your friends to connect with us and hit the like button if you have genuinely liked us. Please also press the bell icon so that you get to know as soon as we come for the next live stream. So take a short break. See you soon. Thank you.